Prova? Cominciamo adesso con le company presentations. Dunque, io sono Susi Longoni di Novava, azienda speciale della NANOFORUM per l'innovazione. Dopo ascoltato che ci siano solo italiani ad ascoltarvi, anche se le presentazioni saranno fatte in inglese. Novava si occupa di innovazione si occupa di fornire informazioni su finanziamenti per l'innovazione ad aziende, centri di ricerca, è partner della rete Enterprise Europe Network, che è una rete della Commissione Europea che ha lo scopo di agevolare il trasferimento tecnologico transnazionale e la business cooperation a livello europeo ed extraeuropeo. Siamo 600 partner in tutta Europa e alcuni paesi stranieri. Operiamo attraverso diverse banche dati contenenti gli annunci di ricerca partner di aziende italiane e straniere e vi invito a visitare il nostro sito che è www.innovav.it per avere tutte le informazioni. L'Innovav si occupa qui a NANOFORUM di organizzare gli incontri one-to-one che probabilmente avete visto su due corridoi più in là. C'è la presenza, grazie anche alla collaborazione con l'ICE, di una delegazione straniera proveniente da Stati Uniti, Canada, India, Cina e Turchia. Procediamo quindi con le company presentation. Dunque, la prima azienda purtroppo non c'è, la dottoressa Lever non è ancora arrivata. Passiamo subito al secondo intervento, quindi Apple Research.
Thank you for your participation and thank you also for this session to make and my presentation will be a short company presentation about the role of Apple Research in developing nanotechnologies. First of all, who is Apple Research? Apple Research means Applied Physical Engineering Research. Is a high-tech company. We are specialized on production of the advanced instrumentation in the field of nanotechnologies. Then we are based in Trieste, in Area Science Park. Area is a big operator, the biggest science park, is a reference for the science park in Italy, and is a multi-sector and technology park, and carries out very types of companies. We can see where we are. Trieste is at the center of the New Europe. Then we are in the center of a very interesting area, and in this center, Trieste was made a lot of investment during the last 20 years, and the result is Area Science Park and also the Cyclotron Trieste, that is a bigger facility that is concentrated in this area. A lot of scientists, universities, and in general intelligence. What is our mission? We have the mission as a spin-off of this university of research center to produce the state-of-art technology for everyday and also advanced application. Then in practical we design, develop and produce advanced microscopy and spectroscopy instrumentation. We have this specialization, and we use the technology for to produce some particular types of standard high-resolution microscopes. And we are really involved in this production from more or less 10 years, and in these years we have constructed an excellent position in this sector. These special microscopes are used in several fields and disciplines. The application of our products are in material science, in surface science, in biology, chemistry, medicine. And our primary market sectors are technology of materials and biotechnology, and also aerospace technology. During the last year, we have a big growth, and we expanded our action not only in Italy, but also in other countries. From some years, we are present in India, with cooperation with some university. We are also present in Europe, Eastern and Western Europe, from Romania to UK, and obviously Italy. We are expanding our markets. This is one reason we are presenting the company here. We are making a big network, sales network. Our characteristics is not only to sell our instruments, but to make some consulting, some instruction of the user that will be helped by our experience, also because inside our research there are some physics, engineer, chemics, and other kind of specialization that making together is have the results to make this kind of instruments. Then to explain better the instruments, I asked Stefano Prato, that's our R&D director, to explain better some examples in a few minutes. Thank you.
Thank you, Stefano De Monte, for the introduction. Just a quick look to what are our main products. This is the list of the main three microscopes that are a part of a family. The family is so-called scanning probe microscopes, and is composed by the AFM, atomic force microscopy, that is the key tool for nanotechnology. Most of the labs that do nanotechnology use this kind of instrumentation. And we also produce some STM scanning tandem microscope, and that is known scanning near-field optical microscopes. Just want to show you with few slides what are the main fields and the main applications. Let's start with STM. STM was the first one in this family to be developed. So it allows you to make a map of your surface. Sorry about this. The surfaces, for example, this is a surface of gold where you can see atomic terraces of gold coated with some kind of self-assembly monolayer. And this kind of instrumentation really go deep into the nanoscale and allow you to make images at the nanoscale. The bright means 8 is a taller features, and the dark is so this is a really a 3D map of your surface. So what you can distinguish here are atomic terraces. So with this tool you can really zoom in. This is, for example, a sample of graphite. You can zoom in till you end up to see every single atoms, almost every single atoms of your surface. So this is a kind of tool that is a kind of the base of the nanotechnology, because if you want to talk about something that is more than 1 micrometer, the first thing that you have to do is to watch it. And those kind of instrumentation, FM in particular, allow you not only to watch the sample, but also to interact with the sample to, for example, perform some modification on your surface. This is, I skip, this is the working principle of this instrumentation. That's what you can look at. Those, for example, gold nanoparticles. You can produce nanoparticles, but it's important to watch them, to measure them, to see if, for example, they interact with each other. The market one, for example, a 1 micron size nanoparticles, the bigger one means that they're kind of interact with each other and assemble in a bigger scale. For example, you can functionalize some surface. This is a glass coated with some gold and then coated with some sugar. This is the first step of functionalizing your surface to grow cells, proteins, to make all the bionano that you need. And clearly, if you claim that you are covering your surface, you need to check the quality of your surface and the quality of your film, the thickness of your film, and some many other parameters that you can be allowed to watch with this kind of microscopes. And then, for example, you can put some cells, grow some cells on that. With this instrument, it's also possible to watch, let's say, from atoms to cells, from, let's say, 0.1 nanometer up to 100 micron. That's more or less the size, twice the size of the cell. And you have here some example of cells where you can really see in a three-dimensional way to measure some parameter to study, for example, all the structure modification of those kinds of cells. A lot of bio-nano is done with that, and is the key tool. And as I said, you can do more than just watching. Those, I asked to have some meters more. Sorry for. So as I say, this kind of instrument you can watch at the nanoscale, but what is even more important, you can interact and modify the surface to make things. Okay, this is clearly some this writing we did it over a surface of silicon, and the width of this line is around 50, 100 nanometers. You can go inside the nanoscale and interact with your sample. Something new. That's what is in technology. So this is about the FM. It is quite popular. We are one producer. We make the project development and the direct marketing of this kind of instrumentation. There are other competitors from the US, but we are acting in going out of our Italian market to propose this kind of instrumentation. Plus, we have another kind of instrumentation. I'm sorry, but the image doesn't come out here. This non-scanning near-field optical microscopes, where we are really living in the world. This kind of we think that this is really the top snob that is available in the market, and this is a kind of a family that is a little bit different from the previous, to allow you to not only the morphological characterization, but is also able to measure the optical properties of the matter at the nanoscale. That's breaking the so-called Abbe diffraction limits. So you can have optical images, as you see on your left, of some biological sample with a resolution that is well below the micron size, that make it competitive against, for example, you have these images in the right that is a transmission electron microscope. So this is another example, just to show you. On the top we have a cut of a piece of section of a tissue, where you can see some muscles or some lipids structure. That is done with the optical microscope. With the optical microscope, you cannot zoom in too much because of these diffraction limits. Okay. So, for example, the muscle fiber is well known. There are some kind of strip-like structure, like you see there, the bottom image. Those are known only by the transmission electron microscope techniques. And with our instrument, we are also able to see this kind of stuff. structure, but more we are also able to measure the optical properties that was not possible to have access to that before. It's clear that if this tissue, for example, is contaminated with some nanoparticle, is somehow altered at nanoscale size, this kind of technique will really allow you to enter the nanopore. So this is a kind of key basic tool for nanotechnology. And this is the team that was put that, as Monty said, we are a multidisciplinary small company, but young and very willing and very competitive in the work market. Thank you for your attention.
Thank you very much. Now we welcome Mr. Giovanni Tosi.
Good afternoon to all of us. Thank you very much, first of all, for giving me this possibility to be here to present to you the party, which is at the University Research Center. My name is Giovanni Tosi, and I'm a researcher at the university, and I'm talking on behalf of Professor Mandelli, which is the headmaster of our center. This center is working mainly on pharmaceutical technology, so we are going to talk about more in nanomedicine and nano-carriers applied to medicine. I have to say this, the faculty is composed by different and several researchers belonging to different departments, pharmaceutical science mainly, but also chemistry, part of earth science and morphological science and anatomy. So it is a multidisciplinary research center. So my job today is to show you, in a few minutes, our research. This can divide and summarize our research in traditional technologies. A few words about these traditional technologies because they are well known. For example, we have skills in matrix and reservoir drug delivery systems for example, localized drug release, laminated polymers for example, a transdermal route, or cyclobestrin complexes for oral administration. But in this NANOFORUM, I would like to talk about nanotechnologies because they are based mainly on liposomes, polymeric nanoparticles, and solid lipid nanoparticles, which are nanocarriers. And we'll talk about them later. Amongst the innovative technologies, we can also produce and prepare microemulsions, for example, for parenteral administration, polymeric prodrugs, so drug polymer conjugates, and we have skills in organic synthesis, also in drug characterization. But talking about nanocarriers, and we apply nanotechnologies to the drug delivery, and we have, I think, one mission, first of all, to improve the patient's compliance, and how we can improve patient's compliance in future application by improving pharmacological activity of the drugs. All of us know about the limits of conventional therapies, the side effects, so we would like to give more selectivity of action of these drugs by specific drug delivery to the real site of action of the drug. So in the fact we are planning, preparing these nanocarriers to reach different goals. First of all, I think, try to load these nanoparticles and nanosystems with hydrophilic and lipophilic substances, which are difficult to be delivered into the body and administered also. We would like to deliver gene. It is great potential application of gene therapy, but gene materials is so much difficult to be delivered into the body and to the correct site of action. We would like to obtain sustained drug release of substances, and we would like also to obtain a stabilized formulation in the body. Stealth liposomes are examples, but also pegylated nanoparticles. In a word, we would like to modify pharmacokinetic values of drugs by enhancing bioavailability of these drugs. And also we have to face also against different barriers, physiological barriers. The first is cellular membranes, but also, for example, the blood-brain barrier, which is a great defensive system of our body, but for drugs it represents really a huge problem and limitation. So let me show our nanocarrier proposed and our skills in preparation of these nanocarriers. The first is polymeric nanoparticles. They are made by polymers, biodegradable polymers, FDA approved. They can incorporate both hydrophilic and lipophilic drugs, and depending on the mode of preparation of these systems, we can obtain an internal structure that could be matricial or reservoir system. These nanoparticles can be stable in the bloodstream, but can be also enhanced in their stability in the bloodstreams and can control the release of the drug. But I think the most innovative thing is to be able to create ultra-specialized systems by means of engineering of the surface of these nanoparticles with proper chemical reactions. The liposomes surely are much more biocompatible than nanoparticles in the body because they are made by lipids, but they show less stability in the bloodstreams. So in the fact, we are preparing liposomes stable in the bloodstreams by means of conjugating with, for example, polyethylene glycols, so stealth non-circulating liposomes. And the other feature that I would like to mention is that these liposomes can encapsulate, protect, and deliver gene materials. So they configure themselves surely as non-viral vectors for gene therapy. And also they can be engineered their surface with ligands. The fact that they are prepared immunoliposomes, meaning liposomes conjugated with antibodies to create targeted and ultra-specialized, once again, systems. And finally, so the lipid nanoparticles are made both with polymers and both with lipids, so a flexible composition. Once again, these solid lipid nanoparticles can be loaded with hydrophilic and lipophilic drugs. They take advantage by not using any solvent, so for a company, for an industry, this is an advantage, of course, and they show a feature which is much more important, this high long-term stability by freeze-drying, and they show also good cell internalization. So to create all of these nanocarriers interfarty, we need and we have skills in chemistry for the modification of the polymers and the lipids, skills in technology, pharmaceutical technology, to be able to prepare these long circulating and controlled systems, and also skills in targeting field to obtain ultra-specialized systems by means of engineering with the proper ligands against a TCO or a CHAL. All of these systems need to be characterized in a different point of view. First of all, the physiochemical characterization, so NMR, DSC, mass, and interfarty, all of these instrumentation are disposable, but also from a technological point of view, in the manner of their morphology, for example, and we use microscopies, so scanning electron microscopy, transmission electron microscopy, atomic force microscopy, as we've seen before, but also we are able to characterize in their size, distribution, and surface charge, which are fundamental and pivotal in the evaluation of the behavior and or the fate of these nanoparticles once administered into the bloodstreams. And then another technological characterization is surely the encapsulation of efficiency of a drug by means of different methodologies such as dialysis. And finally, in the fact we are able to evaluate the efficacy of the systems in vivo on rats mainly, and in vitro on different cell lines. So some results very briefly. The first is application of polymeric nanoparticles to the CNS, central nervous system drug delivery. As you know, our brain is well protected by the blood-brain barrier, but this barrier is a huge problem for drugs. So nowadays, invasive therapies are the only possibility to correct and treat these pathologies such as cancer, for example, brain cancers, HIV, DMX, and so on. So we have engineered these systems, these nanoparticles with ligands able to be cross the blood-brain barrier and to bring the drug into the central nervous system. So in this image, you can see, these nanoparticles are red spots near to the cell nucleus in the cerebral parenchyma. Other application, liposomes and solid nanoparticles for cancer therapy delivery. All of us know about the limits in the cancer delivery and the cancer treatment, so poor selectivity of drugs, side effects, and the possibility to apply gene therapy, but the difficulty to have the correct vector to be able to bring the gene into the correct sites. So we have engineered these liposomes and these solid lipid nanoparticles obtaining stealth circulation liposomes and also conjugating them with antibodies. And we tested on different cell lines. This is in the upper, the ODN delivery to cells by liposomes, and in the bottom, the cell internalization by solid lipid nanoparticles. Now, only the application of the characterization methodologies, confocal microscopy, for example, is often used because it gives a great three-dimensional view of the systems. As you know, this is the nucleus of the cerebral parenchyma, and the red spots are nanoparticles. So through this confocal microscopy, we're able to understand the localization of these nanoparticles in the inner side of the cell lines. So, to finally conclude, I've shown you some application of these nanocarriers in pharmaceuticals and in gene therapy, but all of us know about that these nanocarriers can be applied to diagnostic field, veterinary, or nuclear services, for examples. Then, if you're interested, these are our contacts, our sites, and our emails. And I would like also to inform you that the FAC is creating a consortium to apply a P7 proposal project on biodegradable nanoparticle system for CNS drug delivery for the third or the fourth cohort of this FP7. And I would like to thank you for your attention.
I've been told that the first company has arrived, so this is the event we present as soon as possible, I suppose, or after the last company schedule, so at around 4:30. Now we give the word to Mrs. Elisabetta Tadarico from Veneto Nanotech. Everyone, and thanks to the Chamber of Commerce for giving us this opportunity today. My goal today is just to give you a brief introduction of Veneto Nanotech, the Italian cluster for nanotechnology, and just let you understand what we do and who we are. Veneto Nanotech, again, the Italian cluster for nanotechnology, was founded in 2003 by the Italian Ministry for University and Research and by the Veneto region. Why the Ministry chose the Veneto region to establish the cluster of nanotechnology? Because we had a lot of local competencies present inside the universities regarding the research competencies in nanotechnology. Because we have a big tissue of small and medium enterprises that could benefit largely from the applications of nanotechnology, especially on nanomaterials. As you know, I think we have the cluster for glass, for plastic, for the spore system, for clothing, So these are all markets that would largely benefit from the applications. What is the mission of the cluster? The cluster aims at increasing the investments from private enterprises into research projects. Our goal is creating facilities that already exist, like NANOFORUM that was created in 2003, and work on demand of private enterprise, work for developing their projects, their products, and their processes on their special requirements. So private enterprises come to us and demand to our researchers to develop research for their benefits. The second goal is that of developing technological entrepreneurship. That means that we try, and we actually do that, to raise funds for startup teams, for teams of researchers, Italian or coming from abroad, that have developed an innovation nanotechnology and would like to start their own business. For this reason, since 2003, we organize an international business plan competition called the Nano Challenge, and we give each year a grand prize of 300,000 euro to the best project that is presented. The third objective is that of developing international research centers. We have funded so far established 3 main laboratories, NANOFORUM, EDIZIONE, and LEN, that focuses on specific theme of nanotechnology. This is a map of the Italian clusters that were funded by the Italian government in 2002 by the Minister Moratti at that time. As you can see, we have more or less 21 technological clusters in Italy. And the Veneto region was chosen to establish the nanotechnology cluster for the reason that I told you before. The competencies inside the universities and the tissue of enterprises that could benefit from the application of nanostructures and materials. Our stakeholders are half public and half private. Regarding the public sector, we have the Veneto region, the Italian Ministry for Research, the academic board, we have the University of Padova, Venezia, and Verona, the CNR, the INSTM, and the Center for Colloidal Surface Science. We have stakeholders that come from the financial world. We are strongly supported by private banking foundations and also by some big bank groups. And also we have private societies that are our stakeholders, like LaSalle, like Carel, like Dainese, and the Finmeccanica Group. This is the map of the cluster. Today I just wanted to give you the overview. I don't think I won't have time to go in detail into each laboratory, but what I wanted to underline is the first laboratory that was created was NANOFORUM. NANOFORUM is a laboratory, the nanofabrication laboratory of the cluster. We have more or less 40 researchers working there, and the goal of the cluster is technology transfer. So we work on demand, as I told you before, on private enterprise. So we have a lot of efforts to do on the commercial side to attract private and small businesses to invest money on research projects. Sometimes, you know, small and medium enterprises don't have the resources inside regarding the machineries or the money to develop research projects. So they can either use our machinery and rent our machinery and use the laboratory, or our researcher can work for them according to the project that we have developed together. Then we have CIVEN. This is an association between the 3 main universities of the Veneto region, Padua, Venice, and Verona. We organize summer schools and an international master in nanotechnology for higher education for postgraduate students. So we are launching now the 6th edition of the International Master in Nanotechnology, and if you know of anyone that would be interested, the application can be sent until November. We have this international nanotech campus where students from all over the world come here and for summer schools or the master. So also we organize courses for private enterprise on demand of private enterprises for their researchers.
Then second big laboratory that was created is EDIZIONE. EDIZIONE is the acronym of European Center for the Sustainable Impact of Nanotechnology. We had a protest here outside. Actually, this center is devoted to the study of the interaction of nanoparticles on human health, on the environment, the risk assessment also of this issue. So we concentrated on interaction nanoparticles in human health, on the environment, and also we are monitoring the legal issues. So trying to create a legislation, European legislation for the use of nanoparticles, nanoproducts for people who work in nanotech laboratories or for the consumers. The last laboratory was created is LAN. This is a nanofabrication laboratory focusing on nanolithography especially. So all these laboratories are focused on technology transfer. That's the main point. The research is done following the demand of the groups that come to us and ask to perform our research for them, to improve their products or processes. We go on with, these are some of the results. As I told you, Veneto Nanotech was founded more or less 4 years ago. And so far we have founded 3 facilities, NanoFab, EDIZIONE, and LAN. We have established 400 contacts with private enterprise. And when I say private enterprise, I say either national or international. Groups like Alenia, Aeronautica, BASF, large companies like these. We have started 55 projects financed by private companies so far. We have 4 patents. We have organized 5 editions of the International Master, and we have also funded 7 startups, 7 new companies through this International Nanotechnology Business Plan competition. This is a brief overview of achievement. I told you this is the association of the university devoted to higher education. These are some of the fields of research that are followed inside NanoFab, where 22 researchers were employed, and we were funded by the Veneto region to carry on this research project on nanotechnology with 12 million euros. As I told you, we have in Chile 3 universities involved. We are organizing the 5th edition of the Master. 13 research projects financed by SHIPA funds, 20 researchers, 10 stage internships provided, and 4 projects financed by the 7th Framework Program. These are some of the enterprises that have sponsored our master. And this is NanoFab. NanoFab is the first laboratory. We have 50 research orders from private industries. The very first cost of the laboratory was of 60 million euros start for the implementation of the structure. Some of the scientific know-how, the characterization, coating, and materials preparation, we work here especially on thin films, non-structured materials, coating sensors, and also microarrays for genomics and proteomics. EDIZIONE is the center, the European Center for the Sustainable Impact of Nanotechnology. This is a center that has established a lot of collaboration as a European and worldwide leader, because everyone is facing this problem of interaction of nanoparticles and the nanotoxicity. These are some of the main substances that are being analyzed. LAN is the last laboratory that was created on nanophotonic for cleantech, lens, NANS, and nanosensors. And also in this lab, people can come and use their random machinery and perform their research inside. Other activities of the cluster, as I told you before, one of our best initiatives is Nano Challenge, an international business plan competition devoted to the creation of startups, and so far we have established 7 startups. These are some of the results. We have 4 startups, 150 applications received worldwide, a total investment of 15 million euros. So the one interesting number is this one. thanks to this international business plan competition, we received private funding from venture capitalists that have funded the companies and the startups we presented during this event. Another research center will be open soon is NANOFORUM, that will focus on nanocomposite and polymeric matrix. This will be located in Vicenza. And finally, as I told before, the International Campus for Nanotechnologies, where we have this international master and summer schools. So, to conclude, there should be a lot of things to talk about to let you understand what are our fields of activities. Tomorrow there will be the speech of the scientific director of NANOFORUM, so if you want to come, tomorrow we'll be here in the afternoon at 2 o'clock, sorry. If you want to understand better the field of research inside the laboratory. And in any case, if you want to understand better some of our activities regarding startups or regarding the laboratories, the research, I would invite you to approach me afterwards. I will be here in the afternoon and also tomorrow, and I will give you a larger explanation on each of these items that I can just introduce today. Thank you very much for your attention. Good afternoon, my name is Isabella Cochaco and I'm in charge of the promotional and marketing aspect of TETIS. TETIS is a technology development company active in the emerging fields of nanotechnology and biotechnology, and is developing a new product based on nanostructured material. First of all, TETIS offers a new system for the production of nanopowder and nanoparticles, and supports this customer with also with these two systems. Also is focused on the developing of the process for the production of nanopowder and nanoparticles. TETIS also have a night R&D research and supports this customer with the integration of nanostructured material into device. Is located in Milano, and the team is composed of 21 people. 18 of them are working in R&D, and their background is physics, chemistry, biology, and engineering. And this permits the integration of these different expertise for the development of a solution cross-link nanotechnology and biotechnology. Our laboratory is dedicated for the production of nanopowder and nanoparticles and the characterization, and we also have a full equipped biotechnological laboratory for cell culture and for imaging and for the integration of nanotechnology materials into biological devices. would like to introduce our project. It's a system based on a process, used by pyrolysis process, that is a nitrogen temperature gas-based process for the production of a wide spectrum of nanopowder. The starting material is a solution containing a different material that could be simple oxide,